Welcome to the evolutionary change simulation. This simulation has 5 different parts. In Part 1, called Mutations, you'll investigate mRNA sequences and their corresponding amino acids. To begin, select the mutations icon. Note and record the person ID code displayed in the upper right-hand corner of the screen. Observe the DNA sequence and enter the mRNA code. Then select the Check Answer button to evaluate your work. Next, select the correct amino acid for each 3-letter codon. Select the Check Answer button to evaluate your work. Once your code is correct, select the Make Hemoglobin button to view the hemoglobin. Determine if the cells are normal or sickle cells. Select the Main Menu button to return to the main screen. In Part 2, Sexual Selection is investigated. To begin, select the Sexual Selection icon. Next, choose the Meiosis button. Observe the phase of the sex cells. Determine what will happen during the next phase. To do this, select the number of cells in the next phase in Action using the drop-down menus in the middle of the screen. Select the down arrow to have your selections evaluated. Continue this activity until the entire process is completed. Then, choose the Main Menu button to return to the main screen. In the next section, you'll complete a monohybrid cross. Select the Cross button, then the Dominance Type button at the top of the screen. Choose the question mark on both parents and then enter their genotype based on the information presented at the top left-hand corner of the screen. Select the Check Answer button to evaluate your answers. Select the correct phenotype for each parent. Select the Check Answer button to evaluate your answers. Select the New Cross button to complete a different cross or the selection screen to complete a dihybrid cross. To do a dihybrid cross, select the Dihybrid Cross button. Select the question mark on both parents to enter their genotype and phenotype based on the information presented in the top left-hand corner of the screen. As usual, select the Check Answer button to evaluate your answers. Click the question mark on the offspring phenotype to enter their phenotypes. Again, check your answer by selecting the Check Answer button. Return to the main menu by selecting the Main Menu button. In Part 3 of the simulation, genetic drift is investigated. Choose the Genetic Drift icon. Select the Chance Event button. Calculate and enter the percentage of the initial population after the event. Choose the Check Answer button to evaluate your answers. Next, select the Reproduce button. Calculate and enter the new percentage populations in the first generation area. Repeat this process for the next 2 generations. Again, select the Main Menu button to return to the main screen. In Part 4, Genetic Flow is investigated. To begin, select the Genetic Flow icon. Calculate and enter the percentages of the initial populations of each genotype. Choose the Check Answer button to evaluate your answers. Select the Add Newcomer button, then the Reproduce button. Calculate and enter the percentages for the first-generation populations of each genotype. Check your answer as usual. Repeat this process for the second and third generations. Choose the Main Menu button to return to the main screen. In Part 5, Natural Selection is investigated. Choose the Natural Selection icon to begin. When the Start button is selected, 10 blue and 10 yellow dots will appear on screen. You'll drag the target in the middle of the screen that's to the right of the Start button to a dot to remove that dot from the stage. At the end of that section, you'll count and record the number and color of dots remaining on the screen. Select the Reset button to complete another dot removing session with the same background color. Choosing the Environment button presents a different background color to repeat the investigation.